Hello everyone, my name is Munutra Enoch, and this is the continuation of revision PI 110 question 3. They are saying write with the aid of diagram, show how the following are formed: peptide bond alpha 1,4 glycosidic and alpha 1,6 glycosidic bond. So we start. So for peptide bond, that is just the bond that is found in what? Proteins. So peptide bond. So one thing that you should bear in mind is that for this bond to be formed, meaning there were two subunit or monomers called amino acid that combined. So one thing that you are going to do is that you're going to write the monomer of what? Of proteins where the peptide bonds exist. So we have the amine, they are having carbon with the functional group R there and the hydrogen ion, also the carboxyl group, like that. So what you are going to do is that you are going to write a display structure so that to help us on how the water molecule they are going to be eliminated by the process called condensation or dehydration process, where the water molecules are lost. So I'm going to write any with those we are having to hydrogen. So I'm going to put one hydrogen there and the other one they bonded with what? Carbon with the functional group R there, hydrogen, then carboxyl group, reacting with another one. So hydrogen being bonded with the nitrogen there, carbon functional group, hydrogen, carboxyl group. So what will happen is that the process called condensation is going to occur, where that water molecule is going to be what? Lost. So H, we are having to H that one and that, so H2 and oxygen. So water H2O is what? Lost. So from there you're going to connect these two. So you are having hydrogen N, hydrogen, carbon, functional group R, hydrogen, so C, the double bond minus N, that is hydrogen R with the functional group there R, hydrogen, hydrogen. So one thing that you should bear in mind is that this bond that we have formed is what we are calling as a peptide bond. This bond that are formed there. So imagine, one thing that you should note is that the position of hydrogen does not matter. So we proceed to the next one. So you have to know first the thing, alpha structure. So it is an exos sugar, so that is alpha. So one thing that you should bear in mind is that on prime 1 the OH, meaning the hydroxide, is below for the alpha and the hydrogen on top there. Very important note. So on prime 1 the OH is below, so OH there, hydrogen, hydrogen, OH, hydrogen, CH2OH. So what will happen is that for us to form that bond, it means that they reacted with another alpha glucose sugar. So bear in mind, on prime 1 there is supposed to be hydroxide below, hydroxide there, hydroxide there, hydrogen, hydrogen, hydroxide, hydrogen, CH2OH, like that. So for that bond to occur, it means that the process called condensation or dehydration has to take place, where the water molecule is going to be eliminated. So what will happen is that hydroxide there and that one, they are going to be lost. So H2O is lost, and then from there just connect the two. So we are going to have OH, CH2O, H, OH there, OH there, hydrogen, hydrogen. So there you have it, hydrogen. So they are going to connect that hydrogen. There, hydrogen, OH, 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 hydrogen, 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 CH2O. So this is what we are calling 1,4 alpha bond. The reason is very simple. This is prime 1, 2, 3, 4. So this prime 4, and then this is prime 1. So it's 1, what, 4. So 1, 4. The reaction is taking place between prime 1 and prime 4. So we proceed to the next one. So from there, what you're going to do is that you want to form 1,6 alpha. So you have to draw an exos sugar, which is alpha having hydroxide on prime 1, also that side. So this is prime 6 here, and then that is prime 1. So what is going to happen is that condensation process is going to take place, where the water molecule is going to be removed. So H2O is lost. So from there we are going to remain with so that is hydrogen, there being bonded to CH2, having that exo sugar, so OH, hydroxide, hydrogen, OH, hydrogen, OH, hydrogen, OH, hydrogen. So OH, hydrogen, CH2OH hydrogen. So this that you are seeing there is what we call alpha 1,6, because this is prime 1 and then this is a prime 6. So this bond is what we call the alpha 1,6 bond. So we proceed to the next one. So they are saying state the differences between alpha and beta glucose, also transport RNA and ribosomal RNA. So we start. So one thing that you should bear in mind is that alpha glucose there, so hydroxide is below the ring on prime 1. Now on beta glucose you find to say that hydroxide is above the ring on prime 1. Another thing that we can talk about is that this alpha glucose, they are used as the energy storage. They store energy in form of starch that is in plant, e.g. starch or glycogen in human bone, and then that one it is used in structural support, in structural support, e.g. cellulose. So from there we can go to RNA and tRNA. So the role of this one transport is that they carry what? Amino acid to ribosome. During translation, this one forms ribosomes. Another one is that this one is found in cytoplasm. So the other one found inside the ribosome. So see you in the next video. Don't forget to subscribe, also like and share.